Okay, everyone, we're going to get started. This is Secretary Christy Sullivan. I'm Secretary of the American Society for Cellular and Computational Toxicology. Thank you very much for joining this webinar today. First of all, I want to let you know a couple of things. The webinar is recorded, and members of the Society will have access to the recording shortly after the program. Also, your lines are muted to cut down on the background noise, and after the presentation, there will be an opportunity for questions. What we ask is that you use the question feature to put your question to me, and then I will read the questions to the presenter. And if you have any technical difficulties or questions, please write down this email address, ksullivan@ascctalks.org, or my cell phone number there on the screen, or you can reach me in the chat box as well. So first of all, I wanted to tell you really quickly about the ASCCT. We're a new small society that has been working in North America to provide a place for scientists and regulators interested in and working on the development and implementation of in vitro and in silico methods to collaborate and work together and learn about each other's research. Your membership in part will support activities like this, the GoToWebinar subscription, and we also publish a newsletter. We have a listserv that regularly sends out information about meetings, grants, new OECD test guidelines, and other updates that might be of interest. And we are also going to be having an annual meeting in October. We have an annual meeting every year. This is our fourth, and this time it's going to be down in Research Triangle Park in North Carolina at the EPA building. And we have a really nice two-day program set up where we have a mix of invited talks, which you can see here on your screen, and talks from the submitted abstracts, which were just recently submitted in July. So we're working on that part of the agenda right now. We have a poster session and a social reception as well. So it's really a nice meeting to talk with people, and we hope you will consider joining us. So today's presentation is given by Mr. Andy Forreryd. He's a PhD student at the Department of Immunology in Lund University, Sweden. And Andy works with the GARD project, developing data-driven and non-animal-based testing strategies for the prediction of skin and respiratory sensitizing properties of chemicals. Andy holds a master's degree in biotechnology and is specialized in biostatistics and molecular biology. And his presentation today is titled Genomic Allergen Rapid Detection, a cell-based testing strategy for prediction of skin and respiratory sensitizing chemicals. And so I'm going to go ahead and give Andy control here.
All right. Can you guys hear me?
Yes.
All good? All right, so let's start. Thank you very much, Christy, for the introduction, and also a big thank to you and to the ASCCT for inviting me to present the Genomic Allergen Rapid Detection assay here. So as you guys heard, the GARD assay is a cell-based testing strategy for prediction of skin and respiratory sensitizing chemicals. So the assay was initially developed at the Department of Immunotechnology at Lund University, and it is now marketed and further developed in cooperation with the company Sensigen. So I would like to take the opportunity today to present the testing strategy behind GARD, and I will also try to explain why we believe that GARD is a promising alternative for these endpoints. So let me just briefly start by introducing the process of sensitization. So as you know, a sensitizer is a chemical compound that is able to induce sensitization. So in most cases, a certain chemical sensitizes either the skin, giving rise to allergic contact dermatitis, or the respiratory tract, giving rise to occupational asthma. So, and just to highlight a few examples of such compound, we have, for example, in skin sensitizers, they are frequently included in additives in cosmetic products and fragrances. And we have specific chemicals such as formaldehyde, geraniol, and cinnamic aldehyde. So these are chemicals that all of us are more or less exposed to during our everyday life. So in contrast to the situation for skin sensitizers, the exposure to respiratory sensitizers, they're more common, limited to occupational settings. And the examples of this type of chemicals they belong to very, very specific classes of chemicals, such as diisocyanates, acid anhydrides, and reactive dyes. So both skin and respiratory sensitization is an increasing problem. So in order to limit the occurrence of allergic diseases, it's really important to be able to predict or identify these possible chemical allergens before they reach the consumer market or the working environment. So this type of safety assessment has historically been performed using animal models, such as, for example, the local lymph node assay for skin sensitizers and the mouse IgE test for respiratory sensitizers. But there is a paradigm shift going from animal testing to in vitro-based toxicology for these endpoints. And in Europe, this has mainly been driven by 4 factors in terms of skin sensitization. So the most important is probably the cosmetic regulation. They recently imposed a ban on the use of animals for safety assessment of cosmetic ingredients. And at the same time, there's also a large capacity problem due to the implementation of the REACH legislation. So REACH requires that all chemicals within the European Union needs to be tested for hazardous effects. So testing all these chemicals will not be possible using the current animal experimentation. And in addition, there's also ethical considerations such as the principles of the 3 Rs, and also, of course, public resistance to animal experimentation is a major driving factor for development of alternative methods. And maybe the most important factor for us toxicologists is that the current animal experimentation, they show rather poor correlation to human experience. And this holds true for both skin and respiratory sensitization. So in order to be able to transfer from the current animal-based system into development of more focused and knowledge-based in vitro systems, it's really important to understand the molecular mechanism behind sensitization. And these events are essentially the same for both respiratory and skin sensitizers. So let me just start by slightly review these events in terms of skin sensitizers. And I will later on highlight some of the important differences and also explain how we utilize these differences in order to generate in vitro assays for prediction of both skin and respiratory sensitizers. So the first step in this process is, of course, the exposure to the sensitizing compound. So in order for the compound to initiate the next step, you need to penetrate the skin to get access to the dermis. So the next step of this process is that the chemical must interact with endogenous proteins in the skin to become immunogenic and be recognized by the immune system. So this protein chemical complex then activates keratinocytes, and the activated keratinocytes starts to produce cytokines and other danger signals that helps to establish a local inflammation in the tissue. So the fourth key event is the activation of the dendritic cell. So the dendritic cell takes up this chemical protein complex, and as a response to the danger signals sent out mostly from the keratinocytes, they start to mature and migrate to the draining lymph nodes. So in the lymph node, the dendritic cells present antigens to naive T cells. T cells become activated and starts to proliferate. And upon re-exposure to the same chemical, the allergen-specific memory T cells will be activated and induce a state of inflammation. And this inflammation will recruit various effector cells, which will result in tissue injuries and also development of those symptoms that are associated with, for example, allergic contact dermatitis. This knowledge on how the sensitization occurs forms the basis for development of the animal-free toxicity testing system for skin sensitization. So this table summarizes a few of the assays in development. And note that there is actually assays covering every step in the sensitization process. So we have, for example, the DRPA for measuring peptide reactivity. We measure keratinocyte activation, DC maturation, and also T cell activation. So these are examples on different types of in vitro testing system. But let's move over to and see what we do with the GARD assay. So the GARD assay aims to reflect the DC maturation step, and we believe that dendritic cells are obvious targets for assay development in terms of skin sensitization, and this is mainly because they have a really important role during initiation of those immune responses that actually drives the sensitization process. So the hypothesis behind GARD is based on 3 assumptions. The first one is we believe that DCs will respond differently when they are exposed to a sensitizing chemical in comparison to exposure to a non-sensitizing chemical. And we believe that only the sensitizing chemicals will trigger the switch from DC from antigen uptake to antigen presentation. And we believe that we can use gene expression to measure these changes. And finally, we believe that this gene regulation can be used as a predictive tool to determine sensitizing properties of unknown testing compounds. So there's of course problems associated with constructing assay based on dendritic cells. So normal primary dendritic cells, which you isolate from humans, they're not very suitable for implementation into the assay format. And this is mainly because they are very scarcely distributed in vivo. So it would be really hard to isolate large quantities of the cells in order to scale up the assay format. And they're also normally associated with really large donor-to-donor variations. And that would, of course, affect the ability to develop a robust assay. So in order to circumvent this problem, one can use a cell line with dendritic cell characteristics. So we believe that the selection of a cell line that really mimics primary dendritic cells is a really important aspect for the GARD assay. So we started out the GARD assay project by carefully comparing different cell lines, and we found out that a cell line that is called MUTS3 actually mimics primary dendritic cells, both in terms of expression profile but also in the ability to activate specific T cell responses. And in addition to this, having a cell line which is really easy to grow, that will provide a constant supply of cells, meaning that you can scale up the format of the assay to high throughput. So for basing the GARD assay on the MUTS3 cell line, we have developed this testing strategy for screening of sensitizers. So the concept of the assay is really easy. So we expose our MUTS3 cells to an unknown testing compound. We perform a quality control of the cell. We measure the induced gene expression using complete genomic arrays. We compare this expression to a genomic biomarker signature and to reference sample. And we use advanced bioinformatic tools in order to determine if expression profile of this unknown compound resemble that of a sensitizer or a non-sensitizer. But let me go through this a bit more in detail. So We store the cells in aliquots, which are frozen in liquid nitrogen, and around 2 weeks before we would start an experiment, we take them out, thaw them, and start to expand them. So the cells are really easy to work with. They're doubled approximately every third day, and we only use normal cell medium, which are supplemented with fetal calf serum. So it's only normal medium. So prior to each stimulation, we phenotype the cells by measuring common dendritic cell maturity markers using flow cytometry. So we do this in order to ensure that our cells are immature and guarantee that they are actually able to process and take up antigen. If the quality control looks normal and the cells are immature, we can use this batch of cells for stimulations. So we stimulate our cells with one single concentration, which we call the GARD input concentration. So this concentration is determined according to the schedule here in the red box. So we start out with saying the maximum concentration that we ever use is 500 micromolar. But however, if the compound is cytotoxic, we use a concentration where a maximum 10% of the cells will die. So this is what we refer to as the RV90 value, meaning that we have a relative viability of our cells, which corresponds to 90%. And the third one is if we have a compound with very low solubility, we use the highest soluble concentration. So I would like to demonstrate this determination of the GARD input concentration with two chemicals here. So I used 2-NPD and methyl salicylate. So both these compounds are freely soluble at 500 micromolar. So we need to determine the RV90 for these compounds. So we start at 500 micromolar, we titrate the compounds in decreasing concentration. And for each concentration, we determine the cytotoxicity in relation to an unstimulated control. And we do so by using flow cytometry and PI exclusion. So in the figure down here, we plot the RV90 value or the RV values for the two compounds. And as you can see that methyl salicylate is not cytotoxic at any of the concentration. So the GARD input concentration for this compound will be 500. While 2-NPD are cytotoxic, and it reaches RV90 at a concentration of 300 micromolar. So we will thus use 300 micromolar as the GARD input concentration. So one might wonder why we actually have a maximum in cytotoxicity, why we don't stimulate our cells always with 500 micromolar. So this is simply because we do not want to induce too much cytotoxicity to the cells, because necrotic cells will risk to activate other pathways, for example, response to oxidative stress, and these pathways are not the pathways that we actually would like to measure because they're not linked to the real sensitization process. So when we found out and stimulated the cells with the GARD input concentration, we stimulated the cells for 24 hours. And then we use an in-house core facility for preparation of RNA, for hybridization to the microarrays, and for scanning and data acquisition. So in the last step, we use a supervised machine learning algorithm, which is called support vector machines, to compare the transcriptomic expression profile of this unknown testing compound to expression profile of reference samples. So the SVM classified the unknown samples based on gene information in a reference data set. So the ability of the SVM to classify unknown samples depends on the selection of appropriate reference chemicals to train the model on. So the first thing we did was, for the construction of this predictive assay, was to select reference chemicals. So we selected a total of 20 well-characterized skin sensitizing chemicals and 20 non-sensitizers. We stimulated our MUTS3 cells, we measured gene expression in triplicate reaction, and we used complete genomic arrays. So if we included all the controls, this total data set comprised 137 stimulation, each with a measurement of 29,000 genes. So our final aim was, of course, to use the information from this gene expression data to train a model that are able to predict the properties of chemicals with unknown sensitizing properties. So for the training data set, we have a matrix which comprises our reference stimulation, and we have our gene expression measurement for each of our stimulations. So in addition to this, the sensitizing properties of all the chemicals in the reference data sets are known to us. So we use this information as an input into the support vector machine. So in this step, we train the machine learning algorithm to distinguish gene expression profiles from sensitizers from the profiles of non-sensitizers. So how do we then classify the unknown samples? So we simply stimulate the cells again. We measure gene expression using the complete genomic arrays. We feed this expression data into the SVM model, and we let the model compare gene expression profile of the unknown testing compound to the reference samples, and then we let the model classify the unknown testing compound as a non-sensitizer or a sensitizer. So there is a large problem with microarray data because you will generate so much information. So we measure 29,000 genes for every sample, and then you will get a problem which is called overfitting if you have too many genes in relation to the number of samples that you use. But if we start to visualize our gene expression data from the reference data set here using principal component analysis, so in the plot we have a total of 137 samples, and each dot represents one sample, and each dot also represents or summarizes the gene expression data from one single sample. So we color the samples here according to sensitizing properties. Red represents a sensitizer, and green represents a non-sensitizer. So what it basically means here is that if two dots are very close to each other in space, they have a very similar gene expression profile. So by just looking at this plot, it seems to non-sensitizers and sensitizers seems to be a very, very homogeneous groups in terms of gene expression. So if we try to build a model on this, we need to make the model very complex in order to separate this group. So I made one example down here of the risk of overfitting. So the model to the right is an overfitted model. So this model starts to describe the random noise in the data instead of the underlying trend in the data. So if we try to fit our model to all observations in our data set, we will not be able to use this model in order to classify new data. So we need to find a way to reduce the noise in our measurements. So what we do then is the first thing we'd like to do is to reduce the number of genes to train the model on. So we do this in two steps, starting from the 29,000 genes, and once again we visualize this process using principal component analysis. So we start first by filtering the genes based on a normal one-way ANOVA, so it's a p-value filtering between sensitizers and non-sensitizers, and we do so until around 1,000 genes remained. And as you can see, this starts to appear two groups. So the sensitizers seems to start to separate from the non-sensitizers, but we can still not get a complete separation between the two groups. So even if we continue to reduce the number of genes by p-value filtering, we couldn't get to separate groups. So what we need to do in the next step, we developed an in-house wrapper method, which is called backward elimination. So this method iteratively removes one gene at a time, and then it constructs a model and evaluates the model, and it takes away one gene at a time. So when we have removed around 800 genes, we could identify the GARD prediction signature, which comprises 200 genes, which achieve a complete separation between these two groups. So this is built on the training data set where we actually know the features or the sensitizing properties of the chemicals. So the question is, of course, if we can use this in order to predict the properties of unknown testing compounds. So the best way to find out is, of course, to generate a test data set with chemicals that were unseen by the model during identification of the biomarker signature. So we selected a total of 37 chemicals. 11 of the chemicals we know the sensitizing properties of, and 26 chemicals were predicted completely blindly by the model, and the sensitizing properties were revealed first after data presentation. So we do the same thing again. We stimulate our cells, measure gene expression using the complete genomic arrays, but this time we filter the genes to contain only the expression data from the 200 genes in the GARD prediction signature. So we used our SVM model, which were trained on the samples in the reference data set in order to classify and predict the sensitizing properties of the test data set. So this prediction process is, I try to illustrate this using this principal component plot to the left. So what's actually happening during the classification is that the SVM maps the training data in space in order to maximize the separation between the two groups based on gene expression from the biomarker signature. So the maximal separation between the two groups is here marked by a hyperplane that actually marks the transition between the two groups. So the location of this hyperplane is explained by the algorithm for the SVM. So a prediction of unknown samples are then based on decision values. So the decision values is actually the Euclidean distances from the unknown testing compound to the hyperplane that best separates the training data. So if we use this as the hyperplane as a cutoff, a positive value would indicate that we have a sensitizer, and a negative value would indicate that the model would classify the unknown sample as a non-sensitizer. So we applied this SVM model to predict the sensitizing properties of the 37 chemicals in the test data set, and we were able to determine the sensitivity, specificity, and accuracy of the GARD assay to 92, 83, and 89% respectively. So we believe that this is rather impressive if you compare to the today's standard, the local lymph node assay. So then, of course, one can ask if we actually need all the 200 biomarkers in order to predict skin sensitization. So we tried to create a model which were based entirely on the expression of one biomarker, which is CD86. So CD86 is a common maturity marker for dendritic cells. So we can see that we have a very good specificity for this assay. There was only the sensitizers that induce a significant expression of CD86, but the sensitivity is really bad, and thus also the accuracy of this assay. So this kind of indicates that a single biomarker actually fails to reflect the complex process of sensitization, at least in the MUTS3 cells. So this is the GARD assay for prediction of skin sensitization. So, but based on this, we actually ask ourselves if we could expand the applicability domain of the GARD assay to cover also additional endpoints, such as, for example, respiratory sensitization. So, development of assays for the prediction of respiratory sensitization has proven to be rather challenging. So respiratory sensitization to chemical is rather different from skin sensitization, as I try to illustrate here in this comparison. So first of all, the number of known chemicals able to induce respiratory sensitization are much fewer. Exposure is most commonly limited to occupational system. The target organ is different. They give rise to completely different symptoms. And they seem also to differ in the molecular mechanism that drives the pathogenesis of the diseases. But there is actually two properties of the chemical respiratory sensitizers that makes them a very interesting endpoint for assays built on dendritic cells, such as the GARD assay. So what we find down here is that, for example, the site of exposure. So adaptive immune responses are essentially systemic in nature. That means that you do not actually need a very complex lung model in order to mimic a relevant route of exposure, because skin is also considered as as a relevant place for the priming of the immunological responses. And in addition to this, there's also, even though the mechanism seems to differ between skin sensitization and respiratory sensitization, and skin sensitization is mainly characterized by a CD8 cytotoxic T cell response, and respiratory sensitization is mainly characterized by a CD4 T helper 2 response. The activation of dendritic cells is actually a common feature for both responses. So this made us believe that we actually already had a very good cellular model which were able also to predict respiratory sensitization. So DCs are essentially responsible for driving the polarization of the T cell response into either T helper 1 or T helper 2 response. So given this important role of the dendritic cells, we thought that we can actually use the dendritic cell line in order to identify respiratory sensitizers as well as distinguish them from skin sensitizers. So what we do from here was that we actually started over again. We identified a panel of reference chemicals comprising well-known respiratory sensitizing chemicals as well as non-sensitizing chemicals as controls. So we used exactly the same procedure as we did for skin sensitizers. So we stimulated the cells in triplicate reaction, and we measured gene expression using the complete genomic arrays. So once again, we import this gene expression data into a principal component analysis plot, and here we color the sample according to sensitizing properties. Non-sensitizers are green, blue, respiratory sensitizers are blue. And we started off from the 29,000 genes. We started to filter the genes, once again with the p-value filtering using one-way ANOVA. And as you can see, we can't get a complete separation between the two groups here. So we applied our in-house developed method, again, wrapper method, and we were able to identify the GARD respiratory prediction signature, which actually achieve a complete separation between the two groups. And this signature comprised 389 genes. So as you can see, there is a really good separation between the two groups. But once again, this is based only on the data that we use to train the machine learning algorithm. So this is based on the training data. So once again, we need to validate this data by taking up a new test data set. So we selected a total of 19 non-respiratory sensitizing chemicals and 6 respiratory sensitizing chemicals. We stimulated the cells in triplicate reaction. We measured gene expression using the complete genomic arrays, and we filtered the data to contain only the 389 genes in the prediction signature. So once again, we imported gene expression data into the PCA plot, and the reference data set with the 389 genes was used in order to construct the PCA space, and all the testing compound was plotted into this space without changing any parameters. So it's only the test data, or only the training data set that construct the components of the PCA plots. So this is how the space looks for the SVM model when it starts to classify the samples blindly. So the gray dots are actually the unknown testing compounds before we have revealed the true identities of the samples. So after that, we classify the samples and provide the output. We identify the true identities of the sample, which are here colored in slightly darker color. So the test data set, the non-respiratory sensitizers is very dark green, and the respiratory sensitizers in the test data set are very, they are the purple ones. So if we remove the testing data set, it's maybe easier to see here as well that we could actually have a very good separation between respiratory sensitizers and non-respiratory sensitizers also in the test data set. But we need to validate this using the support vector machine model. So we trained the SVM on the respiratory training data set, and we applied this model in order to predict the features of the chemicals in the test data set. So let me just briefly remind you that the SVM decision values corresponded to the Euclidean distances between the sample and the separating hyperplane. So a high value would indicate that it's a respiratory sensitizer, and a low value would indicate it's a non-sensitizer. So if we then plot the SVM decision value for each stimulation and then color them according to the sensitizing properties, and we can see that the respiratory sensitizers almost exclusively were assigned a higher SVM decision values in comparison to the non-respiratory sensitizers. So the ability of a binary classifier to discriminate two groups that could be estimated using an area under the ROC curve analysis. So a value of 1 in this analysis corresponds to a complete separation between the two groups. So our GRPS reached a ROC value of 0.97, which actually is a very good indicator of the strength of our biomarker to distinguish respiratory sensitizers from non-respiratory sensitizers. And as you can see here also in this plot, that there is only a slight overlap where the model actually classified a few of the non-sensitizers as respiratory sensitizers. So we have a few false positives here, but we don't have any false negatives. So if we once again use Cooper's statistic, we could estimate the sensitivity, specificity, and accuracy of this model to 67, 89, and 84% respectively. So this is so far the highest accuracy of any predictive model in terms of respiratory sensitization. So let me just briefly remind you what we have here. So we have identified 2 transcriptomic biomarker signatures for classification of skin and respiratory sensitization. We have also validated the predictive performance of the signature using external test sets, and we have shown that the signatures were able to predict sensitizing properties of unknown testing samples with accuracies of 89 and 84% for skin sensitizing and respiratory sensitizing chemicals, respectively. So we have now combined these 2 prediction signature into one single assay, which is called the GARD assay. So what we actually do is we simply stimulate our cells with unknown testing compound. We measure gene expression using our complete genomic arrays. We filter gene expression data to contain the biomarker signatures in both skin and respiratory signatures. And we use an SVM model to determine if the testing chemical is a non-sensitizer, a respiratory sensitizer, a skin sensitizer, or if it's even both a respiratory and a skin sensitizer. And this we can do in the same sample. So, in addition to the high accuracy and also the simplicity of the GARD assay, what are actually the largest benefits with GARD in relation to other assays? So I believe that the use of biomarker signature makes GARD a very flexible assay. So, so samples that we test as a test data set, they can later be included also in the reference data set and also be used in order to train the model in order to cover a larger chemical space. So this means that the assay have the possibility to constantly improve its performance as long as we're all the time analyzing new and unseen samples. And secondly, I believe that transcriptome-wide profiling provides a possibility to perform and study the mechanism of action in order to elucidate specific signaling pathways or metabolic pathways associated with sensitization. And third, by using this completely data-driven approach in order to select the genes in the prediction signatures, that is a good possibility to extend the assay to cover also additional endpoints. So we can do this even if we don't know the specific molecular events that are involved, such as, for example, extend to predict endpoints such as skin irritancy. So this is just a very preliminary data, but when we used the information from the genomic arrays in order to see if we could outline some pathways associated with the sensitization in the MUS3 cell line. So to the left, it seems like the respiratory sensitizers in general activates pathways which are associated with, for example, oxidative stress, immune regulation, or apoptosis and survival, while skin sensitizers mainly activates pathways associated with innate immune responses, such as driving the DC maturation processes and also activation of tool-like receptor signaling. So this is very preliminary data, but it's only to show the possibility of the GARD assay in order to also identify different pathways associated with the sensitization processes, and this is something that we're going to continue to work a lot with. So what can we else do? So we also currently try to expand the applicability domain even further and perform measurement of relative potency. So before we only did the classification, a binary classification, where we classify a chemical as either a sensitizer or a non-sensitizer, but now we would like to extend this in order to also perform risk assessment. So this is very preliminary data, but we're currently working with the development of a model which we try to fit to human skin sensitizing potency. So here we define human skin sensitizing potency from 1 to 6, where 1 represents the most potent sensitizer and 6 represent a non-sensitizer. So this is not a complete, a perfect model yet, but we can see from this figure that there is actually some information regarding skin sensitization potency in our gene expression data, and we were able to subdivide the different type of sensitizers into certain subgroups. So just to summarize the whole presentation here, so the GARD assay concept demonstrates a unique opportunity to simultaneously perform hazard classification of both respiratory and skin sensitizing properties of unknown testing compounds. And the assay is able to predict the sensitizing properties of unknown testing compounds with high accuracy that matches the current animal experimentation. And we also believe that the holistic approach of using transcriptomic biomarkers will provide an opportunity to further broaden the applicability domain of the assay to cover also additional endpoints, such as, for example, the prediction of irritancy and also the relative sensitizing potency of skin sensitizers. And by that, I would just like to round up this webinar. If you have further interest in the GARD assay, or if you're interested in running samples on the GARD assay, you're more than welcome to contact us at any time. And thank you very much for your attention, and I will be happy to answer any questions. Thank you.
Well, thank you very much for this presentation, Andy. This was really very interesting. I'm going to take the chairperson's prerogative and ask a question. I have experience with sort of the regulatory acceptance of test methods and know that, for example, with the test set of respiratory respiratory sensitizers that you had, there were 6, I think, positives, and that is probably not going to be enough for regulatory acceptance. Can you talk a little bit about that issue? I know there is an issue with the availability of true positives, and I wonder if you have thought about that, if you are considering maybe collaborating with potentially chemical companies to try to identify additional positives, or if there are other thoughts you can share about that process or potential.
Yeah, so yeah, we're currently looking for partners also for this in the chemical industry. So we have so for now we've mostly been working with the skin sensitizers, and there we have a really good partner we work with within the chemical industry. but I think like the prediction of respiratory sensitizer is not as, or has at least not been as high priority as skin sensitization predictions. So this is, I think this is from our point of view here at least in Europe, it's not very prioritized, and it's really hard to find people with knowledge about this type of chemicals. And so yeah, as you say, the 6 chemicals in the test data set, the positives, that's yeah, it was really hard to find enough sample. I mean, ideally we should have at least 20 compounds to test the model on, but it's, as you said, it's really hard to identify those chemicals, and it's really hard to find anyone who really knows which chemicals are respiratory sensitizers and not.
Yeah. Okay, so the first question we have from the participants is from Lorraine Twardock, and the question is, has there been any real-time, regulatory agency accepted methods to FDA, and has there been any trials yet to identify unknown compounds for allergenicity? I think that you have sort of answered these questions. I think they were submitted early. So I think the answer would probably be not, this assay has not been accepted, if I can presume. Yeah, but it's in the works.
No, yeah, it's not formally validated yet, but it's under that's a currently ongoing process. So it's submitted for formal validation in terms of skin sensitization, but not yet for respiratory sensitization.
Okay, and the next question is from Barun Bhattarai, and Barun says, "Nice work, Andy. Just wondering if the remaining 200 or 389 genes can be biologically justified and not just computationally by removing them for improving predictions." So I think he's asking about a little bit of what you just showed in the last couple of slides, but I'll let you expound.
Yeah, well, yeah, at least for yeah, we're trying to construct like the pathway analysis also in order to see if there's some biological relevance, and well, it's really hard to see. I mean, it's still an in vitro model, and it's just one cell line, so it's really hard to reflect like the whole complex process of sensitization using just one method. But yeah, there might show that there is some biological relevance with the transcript, but the main priority is just to construct a predictive assay.
Yep. Okay. The next question is, how did you determine the number of genes in the prediction model?
Oh yeah, so that is based on 2 steps. So it's first we do the p-value filtering between the 2 groups, and in the second step, so we can, even if we continue to reduce the p-value very low, the 2 groups, you can't really distinguish the 2 groups. The separation will not get better if we continue to do this. So then we use our in-house developed method, which is a wrapper method. So this one is based on that you remove one gene at a time, and then you validate your model by splitting your training data into one training data set and one test data set that is used for evaluation of the model. So it takes away one gene, construct these data sets, validate the model, and then it put this gene back again, and it takes out another gene, and we do so until we get the best possible separation. So this is like a computer algorithm, and it's an iterative process. It takes a lot of computer time and power.
Okay. Next question. Yes. Did you compare when you showed the comparison of the GARD and the LNA prediction accuracy, was that to the human patch test?
No, so that was from the local lymph node assay.
Right, but how was the LLNA prediction determined?
So let's see. No, so they are first identified from, I believe they're identified from different sources, and human skin sensitization potency is probably one of the. So these are just the values for the predictive capacity for the local lymph node assay, and I'm not really sure how they are annotated, but it's probably a mix of human experience data, what's available in the literature.
Okay.
Yeah.
There's a question about chemicals which are not soluble in the culture medium. Do you have anything?
Yeah.
Did you
So
lipophilic
yeah.
compounds, for example?
Yeah, so that was a common problem for the respiratory sensitizers. They're not very soluble, but for the skin sensitizer there wasn't any problem. All of them were very soluble in both in either DMS or water as we use as the vector, and also in the cell media. But we had a few problems with the solubility for the respiratory sensitizers. So some of them we had to use in rather low concentration, but all of them were soluble, but some of them were soluble to a very low concentration. Yeah.
Okay. Has the GARD assay been tested with mixtures of chemicals?
No, not yet. Only one chemical, and that is catone, which is a mixture of different phenol-based things, but not very complex mixtures. No, not yet. But it's in the pipeline.
Okay. So there's a question about 3D tissue models and wondering if that would help expand the applicability domain and would also or may have an impact on the metabolic status of the compound. Has that been discussed or thought about?
Yes, we're actually currently working with the construction of a 3D model where we're gonna combine the MUTS3 cell line with the lung cells. So we have already planned all the experiment, but we're still waiting for the cell line to arrive. But that is something that we have discussed, and that is our next big project going on.
Great. Let's see here. Same question. And sort of a couple of questions around validation. So the first one is, will validation of the assay, the skin assay in particular, be conducted through OECD, and if not, which regulatory body? And a little bit more about the validation process for respiratory, asking what methods, well, basically, are new chemicals only added to the test set once they've been validated by another method? So, for example, in humans, so I guess it's asking if you have, how do you add something, a true positive to the test set? How do you determine if it's a true positive?
Yes, so the first question, what organ we've submitted the test for, right? So it's submitted to ECVAM for formal validation.
Okay.
And the next question, will you please remind me of the next?
Yeah, sorry, I shouldn't have combined them.
No,
It's basically asking if you are adding new positive chemicals to the test set, how do you know that they are positive? Or is there some other, for example, are they a clinical positive? I think specifically for respiratory sensitization.
Yeah. So yeah, it would only be the chemicals we use. They're like well described in the literature,
Yeah.
and there is of course like borderlines, but we do not want to include them, especially not for training of the model, because we really, if we start to train the model on chemicals that are not well annotated, we will have really big problems in order to validate, or even in order to predict true positives and also true negatives later on. Yeah.
Right. There's a question about cost, and wondering if you can share about, maybe a ballpark of the cost of the assay for skin sensitization.
Oh yeah. I don't really know, because all this. So people can run samples if they wish to do this, but that has to go through the company Sensagen. I'm not really sure how much the price will be yet, but I'm sure if people want to run samples, they can probably give you a really good deal. But we're of course also interested in other types of cooperations, especially from the university side. But yeah, still, I mean, the major cost of the assay today is the genomic arrays. They are rather expensive, but we try to improve also by changing to another platform. So we hope to get a cheaper assay. But if you compare to for example the local lymph node assay, it's much cheaper.
Yeah. Okay. Here's a question about the serum media. Does the assay use fetal bovine serum? I don't remember if you said that or not.
Yeah, fetal calf serum. Yeah.
Okay. And are there plans or thoughts about transitioning to an alternative to that?
Yeah, we're actually just before the summer we started to evaluate other types of media, serum-free media as well, or additives. But yeah, that's definitely in the pipeline as well. But we're working on this. But it's still, I don't know. From our preliminary results, it seems like the media or the serum-free media actually activates the cells too much. But we're gonna try different types of serum-free media as well. So that's something that we do as well. Yeah.
That is interesting. So a question on how reproducible is the classification? Okay, for chemicals that are located very close to the threshold or the hyperplane. So when you repeat a test, how reproducible is that distance? And for borderline chemicals, and does the hyperplane depend on the training set, and does it change over time?
Yeah, so that's a good question. Yeah, so we always do our stimulation in triplicates. So we have at least three samples, and we do it on. So every stimulation is from one single cell batch. So we vary the cell batches between the triplicate reactions, and we classify. There is sometimes normally it's quite easy. They normally cluster very close together, the replicates. So it's very robust assay. But we have so we say that if the chemicals are really close to each other, and at least one of them are above the hyperplane, we will classify the chemical as a sensitizer, because we believe that it's more important, at least from the beginning when you try a new chemical, in order to not miss any true positives. Yeah.
Okay. And does it change over time, the location?
Yes. Yeah, it does. That's also one of the reasons why we want to change from the genomic or the transcriptomic platform as well, because there is a drift in the system, but we're currently running control samples all the time, which we use this in order to so the reference training data set is always the same, but we run controls which are also chemicals from the training data set, and we use this in order to calibrate the position of the hyperplane. So it's not a problem, but we need to run a few extra samples. That's the only problem. But the drift per se is not a problem for the assay.
Okay, great. So I think we have all of our questions answered. If I'll just, oh, so sorry, we have one final question.
Okay.
It's about the Moots cell line and the increasing passages. And so maybe you can talk about whether you have some best practices in terms of the number of passages you will use. And if you can talk about that a little bit.
Yeah, so we always use the cells in the same passage number. So we use them in passage number 6, where we split them 6 times before, and we always seems to get exactly when we phenotype them, we almost always get pretty much the same phenotype for these cells. But we have been growing them up to at least 20 passages, and we haven't seen that much of a change. But for the stimulations, when we actually stimulate the cells with the compounds, we always use them in the same passage number.
Great. Okay, well, I wanted to thank you again for your presentation. And if people have additional questions, they can always email me. You can respond to the emails right from the GoToWebinar, and that will come to me or to Andy. And thank you very much for participating, and I hope everyone has a great rest of your day.
Thank you.
Thank you.